what is gene therapy? So first, we have to say what is a genetic disease. So it's a disease due to a mutation or mutations within the genome, the DNA, that will lead to no gene expression or the production of a non-functional mutated protein, as on the scheme on the right. And gene therapy will consist to correct this genetic disease through the reestablishment of a normal, let's say, gene expression. This may be done through many different techniques, and this will lead, depending on the case of interest, to either the suppression of a gene expression, the increase of a gene expression, very recently the direct correction within the genome of the mutation through gene editing, and maybe for what interests us today for CDS and CTD, the strategies that have been chosen so far is more the delivery of a gene construction expressing the correct non-mutated functional protein. So the way to address these are can be very different, from non-viral techniques to viral techniques, and the goal will always be to be able somewhere, for example here, to transcribe the gene into mRNA, and then to translate it into the correct protein. For gene therapy, the viral vectors are very interesting, as you can transduce with them many different kinds of cells. And there are some different families of these vectors that are used for this, like the adeno-associated viruses, adenovirus, lentivirus, or for example, the herpes simplex virus. To explain how these viruses are used in the lab to develop these tools of gene therapy, you must understand that in fact we divert their true role of infectious virus into a tool that will enable us to give the correct genetic construction to the cells we want to correct. So this virus at the end become non-replicative, non-infectious. They will only transduce once a cell. And in fact, we will replace inside their viral genome by a genetic construction that will have the correct sequence to use, like on the little scheme, the creatine transporter and a specific promoter. And these viruses will use their capsid or their envelope to target the cells and then enter the cells that have to be corrected. For the case that interests us today, the viruses that have been chosen so far are the adeno-associated virus. They are quite good to transduce the brain, the central nervous system, and there are a few serotypes that allow this. They can lead to a long-lasting expression of the transgene, and what is very interesting, they don't elicit immunogenicity or very low immunogenicity, which can be a problem with these viruses. They have one limitation, these AAV, they have a small packaging capacity, so you cannot put giant sequences inside them, but for the CDS it's okay. The size of the genes can be introduced in these AAVs. So how will it work? I hope I can use the laser. You see it? Yeah. So the virus will target the cell. I have lost the pointer. Okay. The virus will target the cells on a receptor, then it will make an endosome, it will enter the cells. Somewhere in the cells, the virus will be delivered. It will be deconstructed, let's say, and its genetic material will go into the nucleus, where it will be, for the AAV, transformed in what we call an episome. It's a kind of circle of DNA that will be able to transcribe the correct genes that we want to transduce. So this will make an RNA, mRNA molecules that will go out of the nucleus and be translated in the correct protein inside the cells. The serotype we will use or we will choose will depend on what we want to transduce, depending on the disease, depending on the tissue or tissues to target. So for CDS, which tissues, organs to preferentially target? Of course, one immediately thinks of the brain. For the brain, for example, AAV9 appears quite good to transduce the brain. It is not good to transduce the microcapillaries at the blood-brain barrier. It crosses the blood-brain barrier, but it does not. It appears not to efficiently stops in these microcapillaries. And maybe for the creatine transporter, that could be a problem. And then the genetic construction we put inside, one has to think of the promoter to use. Will we use a ubiquitous promoter that will be active everywhere or in every kind of cells, or specific promoters if you want to target only, I don't know, only neurons or only astrocytes? The strength of the promoter also is a question to ask. And in our case, how much creatine do we want to recover in the brain, for example? We have already asked this question many times during this meeting. Would a partial recovery of creatine be sufficient to correct or to at least to improve the condition of the patient? For CTD, for example, the cells to transduce would be definitely the blood-brain barrier because the transporter is expressed there. The neurons, the oligodendrocytes, there is always the question of the astrocytes, as at least in physiological conditions, the transporter is not much expressed in these cells. And also we have to ask how to inject this virus, this vector. You can inject in the blood, and this, in these conditions, you may, to reach the brain, you may have a dilution of your vector in the remaining of the body before it reach the brain, and your virus in this condition has to be able to cross the blood-brain barrier. Or you can inject intrathecally, so directly within the brain, and with this, you get rid of the problem of crossing the blood-brain barrier. And we always speak of the brain, of course, for creatine deficiency syndrome. But, for example, the creatine transporter is expressed in many other tissues, and there are some questions and some symptoms that also affect some peripheral tissues. So we also have to ask ourselves, should we transduce, I don't know, the kidney or the intestine? So these are all questions to be asked. For GAMT deficiency, that Jerry will present this afternoon, for example, in the brain, you may transduce the astrocytes because astrocytes express GAMT. Can gene therapy generate toxicity? This is also a very important question to ask. And to analyze first, of course, in the preclinical models. You have to look when you are doing your experiments, whether if we think of the brain cells develop normally, have the right morphology, do not die, have the right function. Do we transduce the gene to correct too much, that could lead to some toxicity if we have too much of the protein transduced and in the correct cells. And again, this question, if we speak of creatine, how much creatine do we recover? And not too much, that could be toxic, but enough to have an effect. And there is also the question of the toxicity of the adeno-associated virus. I said they elicit very low immunogenicity, so this is pretty good. But there have been a few rare cases reported, especially on the liver. And this asks the question of the way to deliver the vector. The intrathecal injections may limit this problem of peripheral toxicity. What to expect with gene therapy? For the CDS, of course, to recover some creatine within the brain that could be controlled by MRS, to prevent or to correct the neurological symptoms, and maybe also other peripheral symptoms, like what we observed, like movement disorders, the weight, etc. To end this short introduction, I will just show you a few images of what can be done with this AAV. These are the first experiments we did with an AAV9 vector, transducing a fluorescent protein, either in green, the GFP, or in red, what is called the M-cherry, either by intravenous injection or intrathecal injection. And you see here in green that the GFP was transduced in the cortex or in axons here, or for example in these Purkinje neurons in the cerebellum. So it is, we can efficiently transduce brain cells with these vectors. So these are fluorescent protein, but as you will see in the next presentations, we can transduce the transporter of creatine and also GAMT. And this is an example in the Purkinje cells. In red, you have the transduced correct creatine transporter. So we are able to do this, and we hope that in the 3 presentations of this afternoon, 2 on CTD by Ludovica and by myself, and 1 on GAMT deficiency by Jerry, you will see interesting results. Thank you for your attention.